So, hello everyone. My name is Chris Petak, and I'm a research specialist in the NMR core facility. NMR stands for Nuclear Magnetic Resonance. So that's here at the University of Iowa Carver College of Medicine. Li-Ping Yu is the director of the NMR facility and will be joining us for our question and answer breakout session after this presentation. Our email addresses are also listed here if you have future questions or are interested in developing a research plan that involves NMR. Our entrance is located in the basement of the Carver Biomedical Research Building near the elevator that's across from the Java House. Now, our facility houses 3 high magnetic field strength NMR spectrometers, and the majority of the services we offer revolve around NMR spectroscopy. We can perform a wide range of spectroscopic experiments to address numerous biological and medically relevant questions. So this includes single and multi-dimensional NMR experiments. We offer full spectroscopic services where we will run our samples and also provide data analysis, interpretation, and modeling. So if you find that your major limitation is in sample preparation, we can also perform the expression and purification of your protein for you. So for researchers who want to perform their own NMR experiments, we also offer assistance and training. Now, because our facility houses several high field NMR spectrometers, we're able to offer a breadth of opportunities to researchers interested in exploring the molecular details of their system and solution. Some examples that highlight our capabilities include protein structure determination, metabolite quantitation, ligand binding analysis. So I've picked a few of these specific examples of NMR research projects to highlight towards the end of the presentation. So right now, we're currently working with researchers from a wide range of departments across colleges of medicine and pharmacy. Our Bruker NMR spectrometers range from 500 megahertz to 800 megahertz. NMR spectroscopy utilizes the fact that NMR active nuclei align when placed in a strong magnetic field. Stronger magnetic field strengths lead to better spectral resolution. At 800 megahertz, our magnet has the highest field strength for an NMR instrument in Iowa. Now, all of our spectrometers have proton, nitrogen, and carbon channels and are capable of performing the triple resonance experiments that allow for detailed studies of backbone and side chain atoms in proteins. Our 600 megahertz Bruker Avance Neo is our newest console, running the latest Bruker NMR software. The probe on the 600 also has a phosphorus channel, which can be utilized for studying nucleic acids or lipids or protein phosphorylation. Both our 800 and 600 megahertz spectrometers are equipped with cryoprobes, which provide up to 4 times enhancement in NMR signal. Our 800 and 500 megahertz spectrometers each have a 60 sample changer, and this is capable of automating NMR data acquisition. This can be a useful tool for screening ligand libraries for binding interactions. Now, when running an NMR experiment, the goal is to acquire a spectrum. An NMR spectrum displays a molecule's resonance frequencies, which appear as peaks. NMR is sensitive enough to detect small changes in the molecular environment of atomic nuclei of NMR active isotopes. So these environmental changes can be a function of the molecule itself or induced by interacting sample components. In this way, NMR spectrum can be thought of as a molecular signature for the molecule. So here I've listed some sample requirements for protein NMR. As a general rule, proteins that are below 20 kilodaltons in molecular weight and can be concentrated to almost 0.5 millimolar work best for NMR, but proteins of higher molecular weights that are happier at lower concentration can often be studied by NMR also, but may require some special techniques. And we can help with that here at the facility. Now, the sample volume needs to be 0.5 milliliters, and depending on the type of NMR experiment, may require isotopic labeling. Additionally, one advantage of NMR is that it's non-destructive. It's a non-destructive technique, and so as long as your samples are stable, they can be recovered and reused. So in the next few slides, I'm going to go over a few examples of NMR results that have come from collaborations with NMR Facility, and any publications resulting from them are noted. Hopefully, this will provide you with some insights into how NMR experiments might be useful to you. So, small molecules in biological samples can be quantitated using NMR methods. In this example, the conversion of a metabolite to product was monitored over time by 1D NMR. You can see the NMR peak of the H3 proton on the molecule decaying over time and corresponding to the loss of metabolite. Comparative 2D NMR testing can be used to assess variability of quality attributes during biosimilar development. So these types of experiments can be run on non-isotopically labeled samples, but when you do that, it does require a longer acquisition time. In this example, 2D proton nitrogen NMR spectrum was able to detect the misincorporation of a D amino acid during process development. Because the incorrect peptide was the same size as the correct peptide, other analytical techniques such as mass spec were unable to identify any differences between the two peptides. So 2D NMR can also discriminate between specific and non-specific binding in protein-ligand interaction studies. In this example, changes in the NMR spectra are observed for proteins amide and methyl groups after a peptide is bound. The specific peaks whose positions shift upon binding were assigned to atoms in the protein, allowing identification of residues involved in the peptide binding interaction. So this was possible because the protein was assigned. Now, triple resonance NMR experiments allow for assignment of proton, carbon, and nitrogen atoms in a protein. Our NMR facility has probes specifically designed for running triple resonance experiments on biomolecules. In this example, the assigned proton nitrogen NMR spectrum allowed spectral information to be mapped onto an existing structure. Now, NMR assignments can be utilized to understand a wide range of molecular features of a protein, from thermostability to allosterity to binding interactions. Resulting NMR data is immensely more useful in the context of a structure, a three-dimensional structure. If no structure is available, NMR can also be used for structure determination. Now, NMR experiments can generate a wide range of molecular level restraints that can be utilized to determine protein structures. In this example, the structure of a protein peptide complex was solved using the techniques noted with green arrows. So here are a few additional examples of NMR and NMR-related projects that we've supported research groups with. Hopefully these examples gave you some ideas of how NMR might be useful for your research endeavors. And back to the contact information. Again, if you have any NMR questions in the future, please don't hesitate to send us an email. And I hope that some of you can join us for the question and answer breakout session.